Good morning, everyone. Last meeting, we tackled about the mitosis and the cell division. Today, I am going to discuss the meiosis and the sexual reproduction. Every living creature on Earth begins as a single cell, and that cell undergoes mitosis, dividing over and over again, with certain cells becoming specialized until eventually you get an organism. But where does that first cell come from? Not mitosis, because cell division begins within an existing cell. Instead, this cell is a product of meiosis and fertilization. Note that for sexual reproduction, meiosis has to occur first. So, what is meiosis? Most eukaryotic species reproduce sexually at some point in their life cycles. Gametes, both sperm and egg cell of opposite sexes unite to form a zygote, starting a new generation of offspring. Gametes are formed from a special kind of cell division called meiosis. Meiosis is one of the key factors that generates genetic diversity in a species population and produces the particular genetic mixes that contribute to evolution. Meiosis is a process of cell division in which the number of chromosomes in each cell is cut in half from diploid to haploid. In most organisms, meiosis takes in two stages known as the first and second meiotic divisions. The phase of meiosis have the same names as the phase of mitosis. However, the phase of the first meiosis are actually very different from the phase of mitosis. Both process starts with interphase, diploid and haploid chromosome numbers. Somatic cells are all cells of an organism that are involved in reproduction. Human somatic cells have 46 chromosomes with 23 pairs. The chromosomes that make up each pair are called homologous. Cells which have all the homologous chromosomes are called diploid, while cells which have only one chromosome from the pair are called haploid. Gametes are reproductive cells, and they are haploid. Meiosis reduces the number of chromosomes. Early on, it became clear that gamete formation must reduce the number of chromosomes, or else, with each successive generation, chromosome numbers would double. The fusion of gametes to form a zygote is called fertilization or syngamy. Meiosis exhibits two separate division processes, the meiosis I and the meiosis II. Meiosis I is also called reduction division. The number of chromosomes divides in half, while in meiosis II is called equational division. Double-stranded chromosomes separate, just like as the mitosis. I will discuss it further as we go over the phases of meiosis. Homologous chromosomes or homologs pair during meiosis I in a process called synapsis. Synapsis, also called synthesis, is the pairing of two chromosomes that occurs during meiosis. It allows matching up of homologous pairs prior to their segregation, and possible chromosomal crossover between them. Synapsis takes place during prophase I of meiosis. Synapsis is the connecting of homologous chromosomes to form a tetrad. Look at the diagram of synapsis. During synapsis, homologs exchange pieces of chromosomes in a process called crossing over. Crossing over is a sharing of genetic material between two non-sister chromatids in a homologous pair. Look at the diagram of crossing over. Note that synapsis and crossing over occurs during prophase of meiosis I. Now, let's proceed to the phases of meiosis. The first one is the interphase. Interphase is the period in the cell cycle when DNA is replicated and the cell grows and performs its other necessary functions. The same process takes place in the interphase of meiosis. The DNA in the chromosomes duplicated so that a cell with 46 single-stranded chromosomes in humans will have a 46 double-stranded chromosomes. Once this has taken place, the cell is now ready to enter meiosis 1. Meiosis 1, also known as first meiotic division, occurs in the sex organs, the testes in males, and ovaries in females, puberty stage. During this process, the chromosome number is reduced into half from diploid to haploid, thus meiosis 1 is also called as reduction division. It is divided into several stages that include prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1. The chromatin shortens and becomes thread-like and forms chromatids which in turn form up in pairs called sister chromatid. Because of DNA duplication that took place in interphase, though there are two pairs of sister chromatids. The sister chromatids contain two strands joined at the centromere, so a pair of sister chromatids would contain four strands. In prophase 1, they pair with chromosomes called homologous chromosomes. These homologous pairs are similar in length and size, as well as the genes that they carry. Once these homologous pairs are together, segments of DNA from one chromatid in the tetrad are passed to another chromatid within the tetrad. This provides for genetically different combination than the original chromatid's head. This appearance of the nuclear envelope and the nucleolus takes place. The cell contains 46 double-stranded chromosomes in it. Metaphase 1. The pairs of homologous chromosomes line up at the equatorial plate and spindle fibers attached to the centromere. The first metaphase of meiosis 1 encompasses the alignment of paired chromosomes along the center, or the metaphase plate of a cell, ensuring that two complete copies of chromosomes are present in the resulting two daughter cells of meiosis 1. Metaphase 1 follows prophase 1 and precedes anaphase 1. Look at the diagram of metaphase 1. The homologous pairs of chromosomes align along the center of the cell, while the spindle fiber attached to centromere. And the center is called metaphase plate. Next is the anaphase 1. The actual reduction division occurs. The pairs of homologous chromosomes are separated and one homologous pair, dyad, moves to each side of the cell. The result is that each side of the cell now has 23 double-stranded chromosomes, reducing the number to half. In anaphase 1, the homologous are pulled apart and moved apart to opposite ends of the cell. The sister chromatids of each chromosomes, however, remain attached to one another and don't come apart. Look at the diagram of anaphase 1. There is a separation of daughter chromosomes and the sister chromatids attached to the centromere. Now, let's proceed to the last phase of the first meiotic division, which is the telophase 1. The nucleolus begins to reorganize. The chromosomes become chromatin, and cytoplasmic division takes place producing two cells. Each cell contains 23 chromosomes, which represent the letter N. A cell containing 46 double-stranded chromosomes at the beginning of meiosis 1 becomes two diploid cells containing 23 double-stranded chromosomes. Each will be passed on to meiosis 2. During telophase 1, the chromosomes are enclosed in nuclei. The cell now undergoes a process called cytokinesis that divides the cytoplasms of the original cell into two daughter cells. Each daughter cell is haploid and has only one set of chromosomes, or half the total number of chromosomes of the original cell. Finally, in telophase 1, the chromosomes arrive at opposite poles of the cell. Interkinesis is a preparatory stage of meiosis 2 that occurs after telophase 1 of the first meiosis. Unlike interphase, the DNA is not duplicated. The diploid cells enter this process and second meiotic division begins. Now, let's move on to the second meiotic division. Meiosis II, or the second meiotic division, still occurs within the sex organs. During this process, the chromosome number will be maintained. The whole process is similar to the process of mitosis. During meiosis II, the sister chromatids within the two daughter cells separate, forming 4 new haploid gametes. The mechanics of meiosis II is similar to mitosis, except that each dividing cell has only one set of homologous chromosomes. Steps of meiosis II. Number 1 is the prophase II. Chromatin condenses and forms 23 double-stranded chromosomes that consist of sister chromatids joined together at the centromere. During prophase II, 4 important steps occur. These are the condensing of chromatin into chromosomes, disintegration of the nuclear envelope, migration of centrosomes to either pole, and the reconstruction of the spindle apparatus. However, centrosomes are not present in all cells. Look at the diagram of prophase II. The spindle fibers reappear and the nuclear membrane breaks down. Second is the metaphase II. The sister chromatids line up at the equatorial plate with the spindle fibers attached to the centromere. There are still 23 double-stranded chromosomes in the individual cells. Now look at the diagram of metaphase II. The chromosomes align along the center of the cell, and that center of the cell is called metaphase plate, and the spindle fiber attached to the centromere. The third step is the anaphase II, where the centromeres divide and the chromosomes separate from each other and move toward opposite ends of the cell. There are now 23 single-stranded chromosomes in each end of the cell, which we call the monad. Look at the diagram of anaphase II. The centromeres divide and the chromatids move to the opposite poles of the cell. And the last step of the second meiotic division is the telophase II. The 23 monads are gathered at the poles of the cell as they begin to form back into chromatin. The nuclear envelope develops, the nucleolus reappears, and the cytoplasm is divided, forming 2 different cells. The cells are haploid cells containing 23 single-stranded chromosomes. Since we had 2 diploid cells that enter this process, the whole process of meiosis produces 4 haploid cells with 23 chromosomes each. When telophase II is over, the 2 cells are entirely separated and their nuclear membranes are fully formed. Meiosis is then complete. Let's move on to the sexual reproduction in animals. The reproductive systems. The sex of an individual might be identified by physical appearances. Gametes of animals develops in gonads. Female gonads are ovaries. They produce egg cells or ova. While the male gonads are testes, they produce sperm cells. But there is a special case when the sexes are not separate, and we call it hermaphrodism, where the individual have both testes and ovaries. Examples are snails, hydra, and earthworms. They have rare self-fertilization. Advantages of sexual reproduction. In a sexual reproduction, there are no variations. While in sexual reproduction, the offspring are not identical to the parents genetically. It generates increasing amount of diversity and variation, and it helps species to adapt better to their environment. The whole process of meiosis occurs within the gametogenesis, or the formation of the gametes or the sex cells. Sperm production, or the spermatogenesis. Spermatogenesis is the origin and development of the sperm cells within the male reproductive organs, which is the testes. Sperm cells are produced within the testes in structures called seminiferous tubules. Spermatogenesis can be divided into 3 phases. First is the proliferation and differentiation of spermatogonia. Second is the meiosis. And third is the spermiogenesis, a complex process that transforms round spermatids after meiosis into a complex structure called the spermatozoa. Look at the diagram of the spermatogenesis. Next is the egg production, or the oogenesis. Eggs in ovaries halted before anaphase 1. Meiosis 1 is completed during maturation, while meiosis 2 is completed after fertilization. Oogenesis is a type of gametogenesis through which ova, also called the female gametes, are formed and the produced female gamete is known as an ovum. In general terms, the female gametes are referred to as eggs, but the word egg can involve various stages of development. Therefore, the significance of an egg varies based on the type of organism. Oogenesis is the process of formation of female gametes. This process begins inside the fetus before birth. The steps in oogenesis up to production of primary oocyte occur before birth. Primary oocyte do not divide further. They either become secondary oocyte or degenerate. Oogenesis occurs in the outermost layers of the ovaries. Oogenesis starts with a germ cell called oogonium and undergoes mitosis to increase in number. Comparison of egg and sperm. Egg is round and unable to move, contains the nucleus and a large amount of stored food. Some eggs contain yolk, while the sperm cells are microscopic, made up of head, middle piece, and flagellum. The head is made up of the nucleus and an acrosome. The middle piece contains mitochondria, and the flagellum allows the sperm to swim through liquids. Fertilization. External fertilization is where the eggs are fertilized in an environment outside of the female body. The only sex organ needed are ducts to guide the eggs and sperm cells out of the body. Fertilization takes place in the water where the sperm cells swim to meet the egg. Example is the spawning frog aplexus. Internal fertilization prevents scattering of gametes. Fertilization inside the female requires a specialized sex organ. The fertilized egg is released in a shell or remains inside the female's body. Parthenogenesis, formation of an egg without fertilization. The stages of fertilization can be divided into 4 processes. First is sperm preparation. Second is sperm-egg recognition and binding. Third is sperm-egg fusion. And lastly is the fusion of sperm and egg pronuclei and activation of the zygote.